Good morning. It's a pleasure to be here, and I will do my best to keep you zipping along here. And congratulations to the organizers of this meeting. Yesterday was just fantastic, I thought. A couple thoughts I'll start just to share with you about yesterday. One, I think everyone here would agree that precision medicine or personalized medicine, as you want to call it, is truly a disruptive change, probably the greatest disruptive change for medicine and certainly in the cancer biology world that we will have in this upcoming century. And the challenge of disruptive change is that it's uncomfortable. We need to rethink how we do our current processes and where it no longer makes sense and how we need to change. As that's a good part of my talk this morning. The other is, yesterday I was taken that a lot of the discussion was non-cancer related, and it was about how we identify the biomarkers or drivers of human disease and how we identify that so we can intelligently create new therapies and achieve better, more efficient care and better outcomes through discovery of the biology of the cancer of the human cell undergoing disease. And I would state that, I would at least put forth that in the cancer world, we are probably a little more down the road in that we now have quite a few identified drivers, molecular drivers of cancer, as well as biomarkers that identify them and therapies that are now available to attack that given biomarker in that given particular type of cancer. And so the challenge that we are now grappling with, the disruptive change that we're now thinking about is how do you more rapidly advance the understanding of what is truly a driver of disease and how do we decide these very, very expensive therapies, what's the best way to use them in a way that speeds along drug discovery much faster than our current drug approval processes would allow us to do if we were to continue doing the same old thing. Also, our last speaker has talked about cancer, and was a great introduction to oncology. I would just emphasize that I believe she was talking about what we call germline mutations in the United States. So these are mutations that occur in every cell in the human body and predispose towards cancer. What I'm going to be addressing is more what we call somatic mutations. These are actually the disruptions that have occurred in what was once a normal cell and now are driving that towards carcinogenesis. And so these somatic mutations are more complicated and they evolve over time. So as the patient's natural history of disease and the interventions that are brought to bear will cause different mutations to evolve in an inherently unstable genome. And you get, again, this issue of pathologic versus non-pathologic type of mutations and the need to distinguish between the two, just as the previous speaker spoke of. So this is now starting to come under U.S. regulatory scrutiny. And so that's what I will next move to. So this is, most of my slides are pretty simple. But this A, it just goes by letters. So A, if you follow this along, is how we still do and used to do a test. So we would do a single analyte or a single biomarker test. So for breast cancer, which is what this slide is using as an example, for many years now we've done estrogen receptor testing, and that's a single test. And there are different methodologies for that, IPOX or other ways of testing for that. We then began to see panels of tests. So as we learned more about the biology of cancer, we realized besides estrogen receptor, there were other biomarkers like HER2. And so the laboratories conveniently linked all these tests together into a suite. But still, these are individual tests. Each test has its own methodology, its own, it's really a separate test. It actually is quite consumptive of biological samples, so it's not the most efficient way. Then we began to see the use of computer models or algorithms that began to sort through hundreds of genes to look for expression patterns that might predict a particularly bad type of behavior in a cancer patient. So for breast cancer patients, there are at least two fairly widely used models that do this. One is based on gene expression on 21 genes, and there's a secret formula, a secret sauce, if you will, that gives a weight value to each of these parameters and comes up with a single score, which gives the physician and the patient some sense of how much risk they are of disease recurrence, and therefore whether chemotherapy should be used or not. So it's driving a clinical decision. And it's become widely accepted. It's accepted by payers, it's accepted by clinicians and patients, but it still involves a judgment call as to how much risk one is willing to take. We're now moving to an age of D, the box D, which is next-gen sequencing of genomic hotspots. So these are hotspots that we've identified as being potential drivers of carcinogenesis, at least in some cancers. And now there are several laboratories that now offer these tests. And it's a bit of a race right now. My test does 300 genes, is better than your test. Well, my test does 350, is better than your test. And they're all very loosely called actionable genes, which if you look at the basis of that determination is often quite thin ice. It may be some preclinical evidence. It may be the fact that there's a clinical trial going on somewhere in the world, and that makes it actionable. So that definition is very troubling to clinicians and to regulators now. And eventually, of course, we'll have complete omics analysis, pan-omics analysis, looking at proteins and metabolites, as well as gene expression. So 3 years ago, in 2012, the Institute of Medicine began to take a look at this issue about what are the guidelines or principles that should underlie the use of omics-based tests in clinical medicine. And you'll notice it says omics-based predictive tests. So, So what's the difference between prognostic and predictive? Prognostic is a test that tells you whether a patient may or may not develop this disease, or may not do poorly or well. A predictive test is a test that goes a step further and predicts that this drug might actually work. This drug might actually help the patient out. And so that's a distinction they're focusing now on, prediction of care. And it's a concern because prediction of care is triggering what we call may overlap with what is called the practice of medicine, which is something that has been reserved for physicians to do. And the idea that tests may drive that decision making is something that the FDA is concerned about. Also, you see in green and blue are the differences between a test. So the two green examples are tests where there's enough space between the two lines that this test actually is useful, whereas the blue ones are meant to convey that if the difference is so small, this really is not a very useful test. So the Institute of Medicine came up with 3 definitions that is suggested be evaluated, or any test be evaluated by. The first is the analytic validity of the test, which simply means is the test accurate and is it precise? Is the methodology well defined and understood, and can you trust the actual data that you're getting? The next is clinical validity, which is what you're measuring actually important at all, have any meaning to human disease, whatever human disease you're talking about. It could be diabetes. But for the cancer world, these are what we call driver mutations. These are the mutations that are actually causing either the cancer itself or resistance to drugs. And then the third is clinical utility. So this concept is, well, it's great that you know that there's a biomarker there. It's great that you know that this is a driver of disease, but can you do anything about it? So does it have any utility? Is there a treatment that you could decide to give or not give based on this test result? So these 3 definitions are important, and they've not been fully appreciated, especially clinical utility. So if the IOM then published this as their conclusion, and you can just see by the complexity of it that there's some danger of stifling innovation here because of all these steps. But it is logical. The first is to in any new test that you have a discovery phase where you're looking at the data. Very often it's a clinical trial data, and you're doing computational science to devise an algorithm that seems to predict your observation. Once that is done, that algorithm is locked down and is not changed, and you test that against a second independent data set to see if in fact it is reproducible, not the play of chance. If that happens, then the test is considered to have analytic validity. It then needs to establish previously what I called clinical validity and clinical utility, and I'll jump to the next slide, which will, oops, there we go. This highlighted box will be blown up now. So the IOM concept is that having determined that information, it would then be evaluated by the FDA for either approval or exemption from the approval process. something that is called laboratory-developed tests, exemptions in the United States. And if that gets through that bar and clinical utility is established, then it will go into practice guidelines and drive reimbursement policy. These recommendations have had some controversy. They obviously threaten innovation if they are put in too rigidly. The FDA did release guidance on what it wants to do, and I'm stuck here. Here we go. In January, and of course there was immediate feedback. Some who thought that this was a horrible thing to do and that it was overregulation and it would stifle innovation for precision medicine. And then in the same counterpoint, the opposite point of view saying this is necessary because we need to protect patients. If tests are put out there and we don't really know their value or trustworthiness, we can do harm as much as we can do good. In fact, we'll probably do more harm than we do good. So both sides of this are being promulgated, and the FDA has not finalized its guidance, but this is what is put out for these laboratory-developed tests. So originally, these were tests that the FDA said, we're not going to look at these tests because they're being done probably at a university. They're being done to study a rare disease. They're not going to be developed on scale. And we trust the universities, so we're not going to involve this. This really isn't a threat to patients. They said the test has to be done in a central lab, at the university lab. It's not selling a test kit. That's one of the other reasons. Commercial companies have jumped on this and said, well, if we just do all the tests in-house in our laboratory, we can qualify as a laboratory-developed test and put our product out without FDA approval being required. And so this is being increasingly used to study common diseases in high volume, which is why the FDA is now rethinking this. It's also concerned that these tests often use complex analytics or algorithms to determine their recommendations. And these sort of so-called black box type analysis are not transparent enough to the physician or user to really understand when they might be working and when they might be wrong. So it's kind of accepting the results and acting on it without really understanding from the clinician's point of view, is this test truly applicable? So that is a big concern to the FDA. The FDA's proposal is to take all currently laboratory-developed tests that are exempted and reevaluate them into high, medium, and low risk to patients. By definition, all cancer tests are considered high risk because of the disease and the implications of treatment. So all tests that are currently exempted will undergo review to see if they are, in fact, deserving of that exemption. And all future high-risk laboratory-developed tests will require pre-market evaluation. The FDA has also not issued guidance, but it has issued a request for opinion about its policy for next-gen sequencing. They've asked specifically, should there be standards for analytic validity coming out of the Institute of Medicine recommendations? How much reporting should there be of test performance characteristics, dealing with issues such as tissue stromal sampling, which is the normal host tissue that comes along with a biopsy and can interfere with your test results. As our previous speaker mentioned, questions of variants of uncertain significance, and is this truly a driver mutation, the clinical validity question, and clinical utility. How should the FDA define actionable recommendations and make sure that the practice of medicine is not being subsumed by tests that people really don't understand? So this is not locked down yet, but there is a bill that is being prepared in Congress now called the 21st Century Cures Act, which is meant to be an act that looks at the regulation of health in the United States and how things might be changed. And so we expect there'll be work coming out during the next few months on this. So now I'm going to shift. Now what if you actually had a test and you believed it and had clinical utility where are you at this point as a physician, as a patient? Well, you're a bit of a quandary because you have a test now that says there may be an actionable mutation. By definition, there is a biomarker that this patient has. There is a drug that is used against this biomarker in some other disease, but you don't have the disease. You have another kind of cancer. So how are you going to get access to that drug? And for the pharmaceutical company, they have a drug, and it's already been approved for another disease. But cancer is 100 different diseases anatomically. If you throw in the biology of it, it's thousands of diseases. So how do you design a clinical trial to pursue each one of these? It's impossible. The bandwidth isn't there. The system doesn't allow that. And so ASCO has thought about how we might think of a system, imagine a system where patients could get access, a limited number of patients. You wouldn't need a whole lot, just maybe 20 patients would be enough to tell you whether in fact there actually was a hot signal here, whether it actually was something worth pursuing or not. So how can we re-engineer our delivery system to allow faster discovery? So what would be required would the pharmaceutical industry providing the drugs for free. Now, these are drugs that they already are manufacturing, they're already selling. There's little cost to them for each additional pill or treatment, and it's not a high cost for them to supply a limited number of drugs for this. But we need access to drugs because it's not going to be paid for without any evidence that it works. We need patients who would agree to take the drug, which is probably not hard to find. These drugs are costing $10,000 to $20,000 a month in the US, and if they're not covered by insurance, there's a clear reason why the patient might want to do that. But the patient needs to allow us to look at the data much as you would in any clinical trial or study. The physician needs to report that data to the FDA and to the sponsor of the study, and it needs to do so in a way where it's clean data. Now, it's not the large amount of data we would require in a clinical trial normally. So this is a small amount of data about toxicity and responses as an exploratory analysis to see whether a full clinical trial is warranted. We would need to have payers, either private payers or government payers, to agree to pay for the costs of doing the treatment beyond the drug itself, which is generally has been so for clinical trials. And then we need someone, probably the American Society of Clinical Oncology, to act as the broker for this. So this is my last slide. We think that might work like this, that genomic profiling can be done by a next-gen sequencing or other manner by a commercial laboratory. And there would probably be some limit or some requirements of which labs had data that we would trust. The patient then would be registered into a national registry that would be run by ASCO. The physician would submit the information, a request saying, I have a patient, here's the test result from the lab, this is the clinical history, can my patient receive this medicine? And if it was a good match, then that would be approved, the drug would be delivered, and the patient could receive the medication. So the step that I left out here is what we call molecular tumor board. So under that approved label. So a molecular tumor board is, would be in charge of matching up the drugs that have been received by a pharmaceutical company with the indications that we think might be worth exploring. So it wouldn't be any disease would be automatically approved, but there would be a pre-list of matches that we are interested in. And it might just take 20 patients. If there were 10 or 15 patients who responded out of 20, that's enough signal. If there's no one responding, then not. There also would be a process so that if a patient had a potential drug interest matching to a biomarker, but it wasn't on this list, there'd be a way to submit the reasoning for that and receive special approval. So who benefits? The patients benefit because they get access to a targeted agent that otherwise would not be available. Physicians receive guidance in terms of understanding if there's an actionable lesion or not and how they might find out. The pharmaceutical industry receives data about whether they should pursue this as a formal indication if there's a strong signal of response. Payers get a better understanding of what actually works and doesn't work, and the regulators have information about what's happening in the real world. Right now, many of these drugs are being used off-label, and the FDA is blinded as to actually what happens to that. So they have an interest in understanding that more. So those are my comments. I'll be around the rest of the day and happy to take any questions from anybody at any point. Thank you very much.